second.
Yeah.
Okay, all yours, Craig.
Okay. So welcome everybody to our webinars. Today we have the honor to have with us Dr. Ben Goult. Ben obtained his first degree in biochemistry at the University of Sheffield in 1998 before embarking on a PhD in the labs of Dr. Tim Norwood, University of Leicester, and Professor Lu Yu Lian, University of Leicester and Manchester. And he did it developing an NMR-based approaches for drug discovery. Following a 2 years postdoctoral position at the University of Manchester, he moved to AstraZeneca, Alderley Park, as a senior physical scientist. In 2005, Ben returned to Leicester to work with Professor David Critchley on the proteins that regulate cell adhesion and migration, in particular the firm domain containing proteins tailin and kindlin that are the key players in integrin-mediated adhesion. In 2014, Ben moved to the University of Kent, where he set up his own group on structural mechanobiology of tailin and how cells can convert physical cues into biological signal and to understand the mechanical basis of memory in the brain. In line with this year, Ben and his lab have developed a revolutionary new theory for how memories are stored in the brain, what they call the mesh code theory. This discovery may be the beginning of a new understanding of brain function and in treating brain disease such as Alzheimer's disease. So today we are extremely excited to have you with us, Ben, and we are looking forward to hearing about the brain as an SD card. Okay, so the floor is yours.
Brilliant. Thank you, Christina, and thank you for the invitation to you and to Andrea. It's great to be talking to you all today. It's a shame I can't come to Chile. It's one of the places I'd love to visit. And thank you for the nice introduction. So as Christina pointed out, this is a long-running research interest of mine since 2006 when I moved to work with Dave Critchley, and we were trying to understand how this molecule in the center of the screen. Do you see my cursor?
No, we don't. No, probably.
There.
Yeah, no, yes.
Okay, so this molecule in the center of the screen is the protein called tailin. And I've always been fascinated by how this molecule functions and trying to understand why it's so complex and why it's so essential to life. So what I'd like to try and take you through today is our experimental research into how this molecule functions, how over the course of our experimentation, we've got to this idea that this molecule can store binary information in the shape of its molecules, and how the shape of the molecules can coordinate and instruct the cells how to behave. So the first half of the talk will be talking about the experimental, and then the second will be presenting this theory which has emerged from this study and from this notion that this binary information, culminating in whether to raise the question and to try and open a discussion on whether our brain might be storing memories in a binary format. And I can't change slide. Okay, so this is a video from Harvard University, but we all know about how the brain functions. We understand it's got into popular science culture, electrochemical signaling going around, trillions of neurons all talking to each other, connecting all of our inputs to our outputs, what we feel, what we see, and then computing and giving the correct output. And these trillions of cells are all talking to each other via these junctions called synapses. And this electrical stimulation triggers this chemical release. And this is all well established. But what's not clear is how this can store information in real time, immediate changes, and how it can capture and kind of, I don't know why I can't change slide, capture that information and what that electrochemical signal is actually doing to change and store it. So what we've been very interested in is, and what we've discovered, and what I'd like to present to our idea today, is that we've discovered this array of binary switches that are built into the scaffolding which holds every one of these synapses together. And that is shown illustratively here. So these synapses aren't, the two neurons are touching each other, but they're not they're held together by this complex scaffolding, which stabilizes and holds them together. And built into that scaffolding, there's a network of proteins which are mechanosensitive, which contain these mechanical binary switches. So what I'd like to do is outline how we've got to this realization that the switches in the synapses and what those implications of having switches in the synapses could mean, and to lead to the slightly provocative idea that this might be a way that information can be stored in a binary format in the brain. Oh, sorry. And this led to this paper, which I put out in February, on the mechanical basis of memory. So going from this notion that we've got these binary switches built into these scaffolding, and how it scales from appreciating that an individual molecule in the shape of it can store information, and that information can control the signaling of that site. And then from there to how this might provide a way to consider a mechanical basis of memory, where the electrical and chemical signaling of the brain is coordinating a complex array of binary switches. So if you're interested, the paper's there. It's the top hit on Google currently for mesh code until someone tips it. But our work focuses on these scaffold proteins, and in particular these attachments which cells make to the extracellular matrix, this complex meshwork of proteins which our cells attach onto. And this is shown here with a really old slide now, but this is a cell which is spread out, and these large yellow assemblies are where this cell's physically grabbing onto that extracellular matrix. And what you can see in red is that those attachment points directly couple to the cell's fourth generation machinery, the so-called cytoskeleton and the actomyosin cytoskeleton. And these attachments are really important, not only for just holding all our cells in the right place, but they also provide complex signals which instruct our cells how to behave. And the list of what they do is shown here. Basically most cellular functions have an adhesion component or a regulation via this mechanotransduction. But what I'd like to just focus on as one example is this regulation of gene expression to show the computational power of these adhesive structures. And this is from 2006 now, this experiment, but it's seminal. It's one of my favorite experiments. And it's from Dennis Disher's lab in the Engler et al. Cell paper. And this experiment, they take a population of stem cells in the same media with the same extracellular matrix. And the only variable in this experiment is that they change the elasticity of the substrate. So they make this matrix stiffer or softer. And a stem cell plated onto those different stiffnesses will differentiate and alter its gene expression and its transcription, what it's reading, and ultimately reprogram. So if you put a stem cell onto a jelly-like substance, then you can make a fat cell or a neuron. If you plate it onto glass, then you can make a bone cell. So these cells are feeling what they're attached to and then trying to differentiating to be more like the cells which would be in that environment. So what that tells us is that these blue squares in this figure are information processing centers, because the cell's feeling and then coordinating the whole of the cell's behavior and the whole machinery to do and differentiate. And if we go into slightly more detail and look at one of these blue squares, and this is a picture from Christina's old boss, Tony Kanchanawan. I still love this picture, it's incredible. But what this shows is that one of these blue squares is actually a complex machine. It's got hundreds of these integrin receptors shown in red, physically attaching onto that extracellular matrix. And then it's got an extensive connection to the fourth generation machinery and the cytoskeleton shown in blue. And the focus of our research is on this computational layer, this yellow and green part. And this is where we think all of the, well, not all, but a lot of the computation which is happening through these adhesion sites is occurring in what was originally called the integrin signaling layer. And if we go into a little bit more detail, zooming in now on a single integrin receptor within that, then here's an integrin shown in two different states. It's got a closed conformation, and it can activate to make this open conformation where it will physically hold on to the extracellular matrix. So these different states have different affinity for the extracellular matrix. And the protein which we work on is tailin, And that's what I'm going to talk about today. This is what I think is the memory molecule of animals, but I'll hopefully convince you that why I think that's the case. But tailin's very important for this process because it can pull on these cytoplasmic tails. So this is the inside of the cell down here, and this is the outside. And tailin can pull on these tiny legs and switch the integrin on or off. So it can coordinate whether the cell attaches or lets go. But what tailin also does is it directly couples the extracellular matrix and the integrins here to the fourth generation machinery, the cytoskeleton inside of the cell. And in this position, tailin can experience these forces generated from outside the cell or via the cell contracting its musculature in essence and pulling via its motor proteins on the end of this molecule. And that exerts force on this molecule. And what's well established now for probably 15 years is that when you pull on a tailin molecule, it's mechanosensitive because it will recruit the protein vinculin. And vinculin is shown in green here. And it both stabilizes the shape of the tailin molecules, but also reinforces the links to the cytoskeleton. So these adhesions can grow and shrink as a function of forces, changing their signaling. But something which is really to stress is that this is what's essentially the core of an adhesive structure. You've got an integrin, a tailin, and an actin with vinculin coming on. But if you look at what these adhesive structures look like in the wild, then there's many different structures which form with many different signaling properties and different roles, all on that core complex of the integrin, tailin, actin. And here, these are beautiful videos from collaborators. This is from Mark Morgan in Liverpool, showing a fibroblast crawling along a surface. And in green, we can see just how dynamic these adhesions can be, forming really big, strong adhesions at the front, contracting and pulling, using the fourth generation machinery to pull forward and then disassemble them at the back. The same core integrin, tailin, actin is also what holds all of our muscles together. This is a video from Guy Tannensath's lab showing a Drosophila embryo just becoming contractile, but the same machinery is in our cells. And here the integrin, tailin, actin are holding this together. You've got the podosomes and the invaded podi, all of these different structures which are known to happen and to have really important cellular functions. And here this is a cell which is going through mitosis. And just prior to that, it's spread out. It's got all of these adhesive structures holding it in the right place. And if you imagine in the context of an organism, our cells are all held in where they should be, doing the job they should. But just prior to mitosis, the cell lets go of nearly all of its adhesions, goes into a symmetrical ball, and then mitosis occurs. So the interplay between the integrin, tailin, actin and the cell cycle machinery is really intricate to enable this to happen, to enable cell division in the context of an animal. And what I'd like to then talk about in the second half is these adhesive structures, which form an essential part of the scaffolding, which holds together all of our synapses. Here we've got the ECM, integrin, tailin, actin, physically holding these synapses together. And so tailin is essential for all of these processes. And our working hypothesis in the lab is that tailin is adopting different shapes and different conformations, which are instructing and recruiting different molecules to make different signaling complexes, which drive those different behaviors. And this is a slide of what tailin looks like in molecular detail. This is from our work in Dave Critchley's lab, and this summarizes an extensive effort from me, Alex Gingras and Neil Bate, 3 postdocs in Dave's lab who were trying to work out what the tailin rod looked like. And so this slide summarizes about 4 or 5 years of our work. But what we find is that tailin has got an N-terminal firm domain which binds to those integrins and actually can coordinate whether the integrins switch on or off at one end. And at the other end, it couples to the actin machinery. And what that means is that between those 2 attachment points, we've got this series of 13 rod domains. And what we've gone on to show, and I'll just show you a little bit of the data now, is that these are 13 binary switches. And once you pull on this here, these are all experiencing forces by the cell pulling on it. And the reason, if we just take how we first came on this 3rd domain, we'll just focus on one for this slide, then this looks like this. It's a 4-helix bundle in the absence of force. It folds up into these helical bundles. But when you pull on it, it transitions between a folded state and an open state. And what's important is if you hold this molecule at both ends, then this state and this state are thermodynamically stable. They're just, so they're persistent. If you hold this molecule in a perfect tether, then these two would behave exactly the same at relatively like 3 to 4 piconewtons of force. But what that means is that you can switch back and forward between these two states using force. You can pull on it and let go a little bit, pull, like by moving the contractility, you can coordinate these switches and turn them into different states. And so what we're now starting to do is, instead of calling them folded and unfolded, this is an O state, this is the resting state, and it can transition to this one state when you exert force on it. And we didn't just come to this as, oh, this is a switch. We discovered this biochemically a few years ago now. And that was because we were very interested in a new tailing ligand, which is called RIAM. But as you're in Americans, more the American side of the, they call it RIAM. But this is RIAM here. But this is a protein which takes tailing to the plasma membrane. It's a RAT1 effector. And what we did when we characterized it biochemically was we could show that it works by binding to the outside of these folded O-state domains. And this turned out to be this is called helix addition, where a helix of one protein docks onto one of these bundles. But this was like set into a cascade of experiments where I became a full bona fide badge-wearing mechanobiologist, because this immediately pointed to how you could convert forces into biological signals. Because what was already known about this molecule was that in this unfolded state, it bound to this protein vinculin. So this is the initial recruitment of vinculin, which was mapped out a few years before. But what we could now see was that the vinculin was being recruited only in this one state, but that was simultaneously severing the link to the RAT1 signaling. So as we pulled on this molecule, we changed the signaling in the cell and the signaling on that individual tailing molecule. And this led to a simple question: how hard do you have to pull on a molecule to get it to change its shape and change its biological signaling? And this led to a fantastic ongoing collaboration with Yan Ji in Singapore at the Mechanobiology Institute, and his awesome postdoc Ming-Chi Yao, who's now just set up his own lab back in China. But we wanted to ask that simple question: how hard would you have to pull on a molecule to change its shape and change its signaling? And the experiments which they do are conceptually very simple. You have magnetic tweezers where we can engineer a single molecule. So this is manipulating one single molecule and watching how it behaves. So you can covalently attach a molecule to a cover slip by its N terminus, and then engineer it such that you can attach its C terminus to a magnetic bead. So now we've got a magnetic bead which is tethered to a surface of a microscope via our protein of interest. And then we can use a magnet to pull on that bead, a tiny little permanent magnet. And by exerting force onto the bead, we can exert force onto the molecule. And we can watch how the molecule behaves and how its interactions behave as a function of forces. And I'm not going to show a lot of the single molecule data, but this is just to give an example, is that as we hold our molecule here, as we start to pull on it by moving the bead away, then we increase the force along this x-axis and the molecule starts to get pulled taut. But as you pull it taut, what you see is you see these big transitions, these steps in this. These are each of these domains unfolding completely independently. So here we're stretching 3 domains and we see 1, 2, 3. So each one of these is a switch which is independent of the others. They unfold and refold independently. And this is 6 cycles. So this is, you stretch it till it's completely spaghetti, let go, and then pull. It immediately resets itself if there's nothing else to limit it. And you can measure this. Each one's got its own, each one's independent, each one's got its own force threshold, so they unfold in a specific order. And using mutagenesis, we could start to stabilize or destabilize these different domains, changing the pattern which they'll unfold and making them harder to unfold and identifying which one's which. And it identified that this R3 domain, the one which I showed previously, going from RIAM to vinculin, is the first domain in tailing to change its shape once we pull on it. So to summarize this, because this is from a few years ago now, this R3 unfolding has emerged as a key checkpoint in adhesion assembly. And to go through that really fast now, because I want to move on to the new stuff, but this is a cell migrating down towards the bottom of the slide down here. And right at the front of the cell, you start to accumulate lots of this protein VAM, because you get VAT1 signaling at the front of the cell. And this brings tailin to the front of the cell. And then you start to want to assemble these adhesions. Tailin unfolds, it can grab hold of an integrin. If you imagine what's happening force-wise, then all of this stage is under low force because nothing's pulling on tailin. It might attach at one end, but it's not at the other. But you get to a point where it attaches to, it activates an integrin, which couples to the extracellular matrix, and it couples to the actin cytoskeleton being generated there. And then all of a sudden you have force exerted on this molecule, which unfolds that first domain in tailin, this R3, breaking the link to the VAM signaling and recruiting this protein called vinculin, which then reinforces this adhesion. And then from there, you start to get these very large vinculin-rich adhesions. And this checkpoint makes sure that this only happens at the right place and right time. And it enables the cell to utilize its environment and its intracellular machinery to instruct its signaling. So having identified that initial mechanosensor, we wanted to move on and see how this all of these 13 switches, have they all got this binary switch-like property? And this is, we got much better at the experiments by then and the engineering of these constructs. And this is what happens if you pull all 13 of these rod domains. You start to see this sort of behavior, which is still mind-blowing now, even like 5 or 6 years on, is that as we pull on this, we see R3 unfold here, switching on the adhesion starts to form. But then from there on, you start to get these steps. Each one of these steps corresponds to one of these switches changing its shape. So each one of these, because they've got ligands binding to the O state and the 1 state, each one of these steps is a change in the mechanical signaling and the signaling of that adhesive structure as a function of force. And that means that what we're seeing is a really complex molecule which is changing its shape and constantly changing the signaling at that specific site where it's located. And if you look at what molecules are recruited, this is an abridged list. We're currently expanding this now on different tissue types. But if you look at the signaling molecules which are coming onto those adhesive structures, then they pretty much cover all walks of life in the cell, from like regulation of the cytoskeleton, transcriptional regulators, connections to the pumps and the channels, kinases, cell cycle regulators, all of these different proteins. So depending on the switch pattern of the tailin, you recruit different molecules which instruct the cell how to behave. So we think that this is actually a series of binary switches in coordinating cell behavior. And if you're looking at a cell, between 1 and 9 of these switches are unfolded at any one time. So we think this is an incredibly plastic molecule which is changing its shape as it goes about its business, especially in a migrating cell. We think it's more stable in more stable cell life. But one of the big questions, and I'm not going to go into this in too much detail, is what are the switches coordinating? So we know that this R3 domain switches on adhesion maturation. That's well established now by us and by other labs. But something which was a big surprise to us was that the R7 domain in the O state, but not in the 1 state, coordinates microtubule targeting to adhesion sites. So via this protein called CANK, it binds onto the side of R7 when it's in the O state, and then microtubules target to the adhesion site. So that's coordinated by the conformation, the shape of the R7 domain. And then what we just published last week is that the R8 directly is interacting with the CDKs, cell cycling dependent kinases, to couple the adhesions to the cell cycle and to link these together. But what we start to imagine is we start to get to this really complex behavior of these adhesions, which look to just contain integrin, tailin, vinculin, actin. But within that, you can recruit hundreds and hundreds, up to 250 plus different molecules have been shown to recruit onto these adhesive structures. And part of that is through the shape shifting of the tailin molecules, where the domains are changing their shape. And in the default OOO state, they recruit one set of molecules. But as you start to pull on this and as you start to recruit other things, then you change that signaling complex, which is forming on that adhesive structure. And as you pull harder, you change it again. And if you let the force go down a bit, then it will, so you're constantly changing the signaling. which leads to this notion that there must be some sort of code underpinning this, because we've got hundreds of different molecules which are coming into this complex. We've got forces which are acting in numerous different ways and with different amounts of strength and frequency and all of this. And somehow all of this is integrated and gives rise to the correct cellular response, because we form with high fidelity as an organism. So there must be some sort of code-like behavior underpinning this. And there must be some way to take all of these competitive interactions and these domains which are binding different ligands depending on their shape and these different forces. And so there must be a code to give rise to this robust signaling response. And that's what we're trying to understand in the lab. We want to understand this Tailin mechanotransduction code. And where this gets more complicated is all of what I've talked about so far is a single Tailin molecule with vinculin and all the proteins coming in. One of the complexities is that this isn't just a single molecule. These adhesions have got hundreds of tailins and vinculins and other mechanosensitive molecules. And they form what looks to be like a meshwork inside the cell, which is where this notion of a mesh code arose, because it's a meshwork code where this scaffolding has got this capacity to change its shape and change its signaling outputs. So depending on the forces and depending on the extracellular environment, this will adopt a certain array of ones and zeros. And then all of these signaling molecules will be recruited in different doses depending on the exposure and the availability of the different binding sites. So you're constantly forming this signaling complex. And what you could start to imagine is that on the bottom of our cell or around the edge of the synapse, but in the context of an endothelial cell, along the bottom of the cell, you've got this constantly meshwork, which is constantly feeling its environment and instructing the cells how to behave. And this is just a GIF, and it's not showing, but you could start to imagine that each of those tailin vinculins is a node in a signaling complex. And you can start to wonder what would happen if we were to exert force onto that, and to ask some really simple conceptual questions is like, what if you applied a great force to such a meshwork? And this is gravitational field waves, but it's the same sort of thing. You've got this nexus, you've got this meshwork of signaling molecules, and then you're perturbing it either slightly or with a large perturbation. If you imagine what's happening at each of those nodes, then as you exert force on it, very specifically at that site, you're changing the signaling of that bit of the cell and recruiting different molecules. And that might be to, if it's damaged, to repair it, or it might just change the signaling and enable the cells to constantly be adapting as a function of their environment and as a function of the signals that they receive. And some more conceptual questions, which are what if questions, is that you can ask two simple questions. Is like, it's not hard to imagine, and it's actually true and shown, is that each of our tissues has got its own mechanical properties, different stiffnesses, different stroma, different ECM, creating different mechanical environments which these cells are then interacting with. So you can easily begin to imagine that the pattern of ones and O's on those different environments will have different mesh code patterns. And then if you couple that to each of the tissues has got its own proteome, they're expressing different cells. It's not hard, different proteins, sorry. And this is just shown here with different slices of a brain looking at the proteome. In red, you've got lots of one protein. In blue, you've not got much of that protein. But if you could imagine that somewhere within this complex array are proteins which are interacting with this mesh code, then within a tailin vinculin memory module, you can assemble lots and lots of tissue-specific, cell-specific complexes. And you could imagine incredible diversity and nuance in what's broadly termed integrin signaling. So what we now start to think is we start to revisit this and think that this integrin signaling layer is what I'd like to argue is actually a mesh code computational layer where these binary switches are instructing the cell how to behave depending on its forces and what's available for it to engage with, and also how it's modified. So in essence, what you could start to imagine is that tailin as a mechanosensor, the classic dogma of what tailin does, it's sensing the extracellular environment and it's balancing that against the cell's force generation machinery. And this is shown schematically here as a tug of war between forces generated externally and forces generated internally. And the switch pattern becomes a function of the two opposing forces. But one of the questions I asked, which led to the mesh code theory, was what would happen if the extracellular environment is predictable? And I don't mean it never changes here, but I mean if it's highly regulated and highly remodeled, so it's always predictable. And this is what the cells do. They spend an incredible amount of effort trying to make the ECM predictable. And if you look at any form of neuronal processes, they all require the matrix to be remodeled. But what you could start to imagine is if the cell's going to all that trouble to make the to remodel the matrix, it might be to create a predictable surface. And if it does do that, then this changes this equation, because the extracellular forces are now predictable. The cell knows what they are because it's enforcing those. So this isn't a tug of war anymore. What we're now looking at is we're looking at a series of switches which are coupled to the cell's force generation machinery. So the cell is now in control of its own switches. It hasn't got to worry about this constantly changing extracellular. If you're in a fibroblast crawling along, obviously these are changing a lot, but if you make it predictable, then you can control these switches. And that's what led to the mesh code theory, is this idea that these switches could be internally controlled in response to cell signaling to change them as a function of signals going through that past to stimulate the contractility. And that's all based on this realization that this is what we're looking at here. And so what we now start to imagine is that putting all this together is that this is actually tailing a data molecule, because we can show, and I hopefully I've convinced you, that we've got a string of binary switches in this molecule able to go between an O state and a 1 state reversibly as a function of signaling. And if you pull on it, you'll change the order, the binary string from 000 to 010 or whatever, in a uniform way, in a highly reproducible way. So that you pull on it, you'll generate the same pattern every time. Then you start to imagine a scenario where each of these molecules in the scaffolding can adopt different signaling complexes. And then as you generate contractility, as you stimulate a synapse, say, and you generate the switch on the motors, you have contractility gets switched on, that's well established, then that pulls on these scaffolds, changing the pattern and changing the signaling output as a function of the signals it's received and as a function of the shape of the molecule prior to that signal. So this is a constantly updating way to send information forward in time, because the shape of these molecules and the signaling complexes which assemble is a function of what's already happened and what's happening going forward. And this can be constantly changing back and forward as a function of alterations in the contractility through these signals. So what I'd like to argue is that this is information that's being stored in these molecules, and we can show that sort of information because here's some, we can now, as we know how these molecules are behaving, we can do this experimentally and in simulation, is that you can physically push and pull on these in a uniform quantized manner. And you can write information into the molecule, into the shape of the molecule as a function of force, adding force, have a contraction, adding it. So you can build up these and write the shapes of these molecules in an incredibly reproducible way. But a big part of it is that it requires that the mechanical environment is defined and predictable, as I've already mentioned. And that leads to a notion that I think, and it's for a long time, mechanobiology or mechanotransduction in the brain's sort of been a bit thought, because the brain's so soft and squidgy, it's not necessarily being considered to be a highly mechanotransductive environment. But what I'd like to argue is that based on this theory and what this theory predicts, is that while the brain's soft and the actual, each synapse goes to a lot of energy to create basically a nest around the outside, this meshwork of ECM, and it's constantly undergoing maintenance and remodeling to ensure that this is maintained. And the prediction of the theory would be that the reason it goes to all this effort is to enable mechanical computation to be perfect. Ooh. Because if you look at, this is a terrible artist impression, which I do. I'm not an artist for any stretch, but this is a synapse just shown schematically here, a pre-synapse, a post-synapse, an astrocyte. If you imagine around the edge of that, you've got this cell adhesion molecules holding it together. So it's not just tailing, there's other cell adhesion molecules, obviously, but together they form this scaffolding holding this together. But in the mesh code framework, what we start to predict is that, or start to imagine, is that the synaptic scaffold is containing thousands of these mechanical switches. And when you have electrochemical signaling and it stimulating the release of the chemical transmitters, and then you stimulate that synapse, that causes contractility on both the pre and the post-synaptic side. And as this musculature contracts, it's like a short burst, it alters these patterns in these switches. So this binary information written into that synapse is altered, is the prediction of this theory based on what I've told you so far. And as a prediction, if you imagine there's 100 tailings, then you might have up to 100 bytes information. We don't have actually actual data, but these switches might enable this information. So as you signal, it captures that information, because these conformational changes can happen really fast. You pull, you can change the shape. So you can actually retain that electrochemical signaling and alter the activity of the synapse. And that's where we come to this theory, this mesh code theory, that what we're actually looking at with the tailing, what we've been striving to understand all that time, is that this is a binary coding, and that the brain is in essence behaving a bit like a mechanical computer, where you've got this mechanical signaling which is occurring, and this electrochemical signaling is coordinating this cytoskeletal machinery, this mechanical computing nature, constantly dialing up and down each of these synapses, making them more active or less active, and capturing that information at the same time. And what's also quite exciting about this theory is it would go from, it could enable computation in the brain, but it could also enable, because all of our cells are attached, they've all got adhesions of some structure, some sort, then maybe all of our cells are running essentially a binary coding where there's the patterns of them are instructing each of the cells how to behave. The computation in the brain sends signals to the different parts of the body, and it can all be written into some sort of binary coding. And a big question would be, how would this be read? I'll skip through this because we've already talked about it in essence. But if you start to imagine a binary coding in a specific synapse, then if you had an ORO state, you might have one set of synaptic regulators, scaffolding proteins, etc. bound. So your readout of that pattern would be one certain signaling complex, one certain activation state of a synapse in essence. You pull on it, you alter the pattern of those signals, and it might be that that then primes that synapse, or it might be that that suppresses that, but you could be constantly tuning the activity of the adhesion complexes in each of these in a much more dynamic and quantized fashion than what you might predict. And you pull on it again, you change it some more. And what's really, I think, is exciting about this is that this is what's been known in the adhesion field for many years, this contractility dependent growth of adhesions, where if you stimulate an adhesion, it gets bigger, it changes its signaling. If you don't stimulate it, it gets smaller, it atrophies. So you constantly, it's a simple set of mechanical rules which would be underpinning this process. And one of the readouts would be that all of these little yellow, green and orange, etc. blobs, proteins, they're all getting relocated to these different switches in different regions of the cell, depending on what patterns are available. And I think my next slide should hopefully show that. I might have crashed it. So yeah, so this is just to try and conceptualize this a little bit in terms of a neuroscientist slant, is how would a mechanical code be read? And what I would like to say is the prediction from the theory is that electrochemical signaling, shown by this lightning, results in a short contractility either side of the synapse, which is well established that you do get activation of actin-myosin contraction at stimulated synapses. And that would lead to changes in the scaffolding patterns. And potentially in the presynaptic side, you might even release or activate or trigger alterations and lead to help to change the probability of the synaptic the release of the vesicles. And in the context of a mesh code framework, that might be dead easy. If there's a tether which is bound to one of these switches, you unfold the switch and release it. That's something which we're actively working on at the minute. But at the other side, something which is really well mapped out, but not really well understood, is this concept of synaptic tagging and capture, where a stimulated synapse recruits lots of newly made molecules compared to an unstimulated synapse. And it's sort of like predicted that there's some sort of tags which are formed which lead to this, but those tags have been elusive. In the mesh code framework, this becomes really obvious because these tags would be the exposure of these cryptic binding sites, which then recruit the molecules to that site, and only the synapse which has been stimulated and had the contractility would have those sites. So you'd recruit molecules to the stimulated synapse by exposing these sites. And the opposite of that's also true, this inverse synaptic tagging, where you have other proteins which are recruited to dormant synapses to keep them in a low unpotentiated state. And in the same idea, then if these are bound to that same switch, then those dampeners would be released and they would accumulate on the unstimulated synapse. So you'd be constantly talking between all of these different synapses, moving these different proteins around as a function of this, and in a very quantized manner. And then later, as you exert more forces on that, you'd be changing these patterns further. And that might be how you could start to have more complex arrangements. And ultimately, my argument is that this is how you write physical memories. This is how you store an engram, essentially, that these patterns of ones and O's and the signaling molecules recruited ultimately give rise to this memory trace. And what time is it? Yeah, 5 more minutes. I'm nearly done, but what I'd like to just say is that this then scales quite nicely because you could have an individual memory molecule, which we know exists, and an adhesion as a memory complex coordinating a synaptic activity. And part of this mechanical computation would see synapses release, stimulated synapses, so triggering whole circuits. And the whole circuit would be activated via this. And then the return back to homeostasis would result in, so when you stimulate it, this is just a neuroscience of a reverberating circuit. But when you stimulate that circuit with an input, all of these synapses would be activated. So you switch on this circuit, and that would lead to altered patterns in all of the synapses and all of the spines and all of the bits in that circuit, ultimately giving rise to an output. And that at the end of that calculation, the synapses would look the same essentially, but the patterns of information in them in the whole circuit would be altered, such that you would have an input stimulating an output, but these patterns would be altered, would be the prediction. And this might mean that the next time you stimulate that, you immediately stimulate an output. It might be that you've learned that behavior, or it might be that these patterns are constantly updating, sending this information in different directions to calculate or compute what is going on. So in this theory, the information would also be controlling the synaptic strength and the probability of firing. So there'll be this direct link between this information and which synapses and which circuits are switched on. And also where I think this fits quite nicely is that this concept of how information is added into these calculations. So I'm sort of proposing here that this is a binary code, it's a computation. And the way that we feed information into that is through our sensory neurons stimulating and action potential spike trains, where they send a binary pulse of all or nothing signals. And in the context of the mesh code framework, then this guy would be putting his hand in the shower. He would detect that, he would send signals to his brain, there'd be computation, but all the way along at all of these synapses, that information would be being updated and these synapses would be changing as a function of that. So it would be doing the computation up here, but it would be constantly updating the whole circuitry of the organism with the information of the best probability by tuning the activity of these synapses via this coding is the prediction of the theory. And what you could start to imagine is you could imagine the entire representation of the animal's unique life in this coding, like, because every single input would then write into this patterns of ones and O's in these synapses. And ultimately, each from a relatively default state, the brain would be updated with all of this information depending on what that individual experiences. So just to finish with a few thoughts is that what we can say with certainty is that we've got memory molecules which can store information in the shapes of them. And each of these switch domains in each of these molecules can store one bit of information. And what you can start to imagine is you could have an array of these switches, and that would enable the storage of vast amounts of data. And if you look at what the cortex looks like, this is one of my favorite diagrams from 20 years ago now, or 15 years ago, whatever, it's 25 years ago. maths. Then the cortex of the brain is incredibly logically ordered. The architecture is fantastic, that you've got these teardrops here, each with 60,000 synapses. But in this concept, you've basically got hierarchical structure of columns, these 2 million of these columns, made up of synapses, made up of neurons, made up of synapses, made up of mesh codes, made up of tailings, made up of mechanical switches. So this is an incredibly hierarchical logical structure, which I think has striking similarities to what we've converged at in our desire to optimize data storage in pen drives and in SD cards and solid SSD cards, where you've got these pages of information with blocks and individual ones and O's, all wired up to these channels, incredibly repetitive logical architecture, all connected back up to the memory module and the memory controller. Same here, this is all connected back up to the hippocampus. So it almost says that from this concept of this binary information, you can start to write information into specific regions of this cortex, almost like we'd write to a disk. So if that's the case, then that would mean that every synapse could be specifically addressed by neuronal activity, which I think is probably true. I think that's what you can see with the electrochemical signaling, which people are starting to be able to see that level of detail. But if you do have that, then one of the major things which follow is that every memory we remember, every bit of information we ever remember is written somewhere into the dark recesses of our cortex. Finding that information, if you try and find information on a computer from 5 years ago, some data, it's really hard. You've got to have a really good addressable system to be able to call that data instantly. So it's a major challenge, which is memory allocation. And this is what's really coming, I think, is really coming, that this is being demonstrated. So this is a beautiful papers from a couple of years ago, where you can measure the electrical fingerprint of a mouse's activities going about its business in the day. It goes to sleep, and then it consolidates all those memories into a really precise set of electrical fingerprints. If you imagine this in the mesh code framework, then this would be constantly updating this binary pattern, moving this data around on this disk, and then the next day the memories are consolidated. So maybe REM sleep, where there's all that electrochemical activity, is the physical moving around and the contractions to change these patterns of ones and O's to write this information. So that's all I wanted to say, but just to ground it back onto, it's experimentally derived this. It's not just based on purely on theory or out of the air. I'd love to discuss this with you all, is that we've identified that tailing's got this molecular memory and that it can store this data persistently as a binary string. And what's exciting to me is this is based on really well-established biophysical and biochemical rules. It doesn't require delving into the quantum world. It doesn't require unexplained forces. This is simple biochemistry, simple behaviors. But what it also means from that simplicity is it identifies, in my opinion, and what I'm arguing here, is a physical location for data storage in cells and in animals and in the brain. And it might be that because all of our cells have got this same machinery, the same mechano-memory machinery, that maybe this is a machine code that's keeping every single cell running in synchrony and working to the same hymn sheet, so to speak. And in the brain, what I also think is interesting is that this doesn't, all that's already known about the brain fits beautifully into this model. Like all of the chemical and ionic signaling layers, which are well mapped out and understood, they would be going on doing their business, but they'd be constantly updating this mechanical layer underneath, which would be the mechanism for the information processing and storage. But what I'd finish with is the conversion and storage of these sensory and temporal inputs into a binary format identifies an addressable read-write memory system. I think you can take all the information from all our senses into a binary format, into a binary calculation. And I think that's important, that you can read and write this. And to finish, I think that this supports the view of the mind as an information processing system and an organic supercomputer. So if you want to know more, then the paper, it came out a couple of months ago. If you search mesh code, you'll find it. But just to finish up, I'd like to thank the people who've helped him with this work. In particular, I'd like to thank Ji and Ming Chi, because we started on this with this simple question, how much force to flick these switches? And it just opened up this huge, every experiment showed us something which we'd never have predicted. And it's been super exciting, and it's a fantastic journey. I'd also like to thank Martin Schwartz in Yale and Nick Brown in Cambridge, because we've had a lot of philosophical discussions on this, what it might mean, challenging the theory and thinking about it. The guys in the lab who are actually doing this work, Dave Critchley, who's been a long time supporter of me, and the guys who have funded this work. And I would like to thank you all for listening, and hopefully there'll still be time for questions. Thank you.
Thank you, Ben. Awesome, really fantastic. While you were talking, I started thinking about neuronal cells as, this old puppet with the string inside, and everywhere you put them, they move. So in that sort of terms, it's really impressive, really beautiful. Okay, well, the other warm-up, I have maybe start with one question, which is, so in the brain, everything is tightly controlled. And so your theory would work for that type of environment. I mean, the neurological properties of the ECM are tightly controlled. Do you have also, or have you thought to expand this theory for other systems where that is not regulated? Like I'm thinking cancer development, where you have a bidirectionality of the informations.
Yes. So that's a good question. So yes, we definitely do. That's where we came from, more of a cancer metastasis type of thing. So one of the things we've just got a big CRUK program grant to look at is that this coding, this behavior, keeping all the cells in check, during a lot of the cancer, like in pancreatic cancer, where you start to stiffen the stroma and you start to change the mechanical properties, that this mechanical coding might be one of the things which ultimately keeps it in check, but then flips and suddenly switches to a different program. So yes, we're incredibly interested in when this goes wrong, and also in the brain, like where this might go wrong in neurodegenerative diseases, where you might start to get this folding and unfolding happening incorrectly. Either it gets too stiff, or it gets clogged up, or it starts to get out of sync. So as a concept, where it's quite nice for thinking about diseases with a different way, like Alzheimer's or cancer, where these things go wrong, and it might be that this offers a new angle to consider those sort of things. So yes, we're definitely thinking about that.
Fantastic.
Well,
so any other questions?
Yeah, me. Well, not a question, more a comment, but well, I'm really intrigued about this binary code associated with tailing, and well, obviously you can think that well, tailing in the focal adhesion, but in the cell adhesion model might have a similar way of working, right? So I was thinking that this mesh code, computational code, also has a layer in the cadherin-mediated adhesion, so with alpha-catenin and vinculin instead of tailing and vinculin. So it would be very interesting to deepen and to follow up in that direction as well.
Yes, I completely agree. And vinculin itself, I don't talk about here, has got a number of these switches. Alpha-catenin has. So the reason for broadening out slightly from the tailing code to the mesh code is that it's almost like the whole of the cytoskeleton is a mechanical computation where they've all got these strings, and the adhesions, the reason for focusing on the adhesions in this is that they've got these 13 switches and they're attached to a knowable surface, which the cell's in control of. But it would be coordinating the cytoskeleton and the, yes, so absolutely. The interplay, and even just the dosage of the vinculin, whether it's at the adhesion, the cell matrix adhesion or the cell-cell. If the amount of vinculin was limiting, then you'd have this crosstalk just by enforcing the switch from that. So yes, definitely.
Yeah, correct. Yeah.
So there's a really nice, I think there's a really nice story to be got at on this crosstalk between the cell matrix and the cell via this coding. So I agree.
Yeah. Thank you. Mauricio has a question. Please open up your mic.
Yeah, just raise my hand. So thanks for the interesting presentation. As a computer engineer, I'm very curious about these theories about how to store memory. So I have two questions, both related with time. So this, how can you compare the time it takes to this vinculin molecule to change compared to the spike in time? So how fast is this compatible in terms of timing? That's one question. And the other question is, you at the end compare with other engineering ways to store data, like magnetic of solid state drives. So all these devices have a characteristic of time. So a magnetic disk will typically last for 8 years, 10 years, depending on the quality. So how long this with this model the memory will stand?
Yes.
Or what it needs to stand?
Yes. So that's so the timing. I mean, once the force exerted on tailing overcomes that threshold, it's instantaneous. But how long to generate that force? How long to generate the machinery? To switch on the contractile machinery? Yes. But the good thing is that once you start that, it's almost like a chain reaction. Once you stimulate it, you trigger a series of events which leads to site contractility, which leads to these patterns changing. So the times are all to be worked out, but they're fast. And if you hold the molecule, then it will never refold. But the trick here, I think, is that the unfolded states are stabilized by the vinculins coming on and off. So you have a persistence to this. And also one of the things which Francis Crick was saying back in the 80s, or was it 90s, about memory in biomolecules is quite hard to imagine because they turn over quite a lot. But here we're talking about a system level thing where we've got these switches which are intimately linked, but it's part of an adhesion. Like on top of these switches, you're assembling like 100 or 200 different proteins. So it's a complex thing. But those signaling machines are explicitly dependent on the pattern of information underneath them. And in terms of the lifetime of a disk, well, maybe the reason now we're living into our 70s, 80s, 90s and beyond, and the memories are starting to fade, is maybe that they're butting up against the lifetime of the disk, so to speak. But the difference between the brain and a solid state disk is that the brain has got the capacity to constantly repair and remodel, and the plasticity and the changing of all of this stuff, for as long as it can maintain repair it enough and maintain that, then it could last. Well, we know how long our brains last, how long you can remember some stuff. So I don't know if that answers the question. But the other, one of the things which has fascinated me is like if you get hypoxic, then within a few minutes your brain gets corrupted very, very fast, even though the integrity of a lot of it doesn't look too damaged. But I think part of the reason is that these switches, they're all talking to each other. So the whole thing is mechanically synchronized in this theory. And if you lose that, if motors in one part of the brain get switched off or something, then that will come out of sync with other parts. So then you start to get like corrupt sectors, or to really labor the analogy, but so that sort of thing. So how long it would last, yeah, I don't know, it's to be worked out.
Yeah, I would say as long as the energy flows in and you keep your mechanical engine going.
I think so, yeah. And then
stuff in that direction.
Yes.
Yeah, Miguel, you have a question?
Well, indeed, I have actually many questions. And thanks for the talk, Ben. It's very interesting, and yes, it's just moving around my mind. Many things you have presented today. So just two things, because I need to digest a bit your theory. So just two things came to my mind when you were talking about, okay, you're centering all, okay, on the ability of the synapse to respond to the biochemical signaling, electrical signaling through contraction of the actomyosin cytoskeleton, yeah? And then interact with the extracellular matrix, and then according to this interaction set states that can be reinforced while the activity of this happens from time to time, okay? So I just wonder whether, I mean, the mechanical properties of the brain, so the softness of the tissue is actually an advantage or requirement for this theory to work, because this provides like a kind of dynamic range of the possible responses to the changes or interaction with the environment. I'm thinking also that the brain, for example, with age becomes more rigid, and in this, I mean, the memory formation is impaired in this condition. So is that a requirement for this tissue to be soft, really, to allow this memory configuration? This is one question. And the other is just a comment about, I mean, if you can make a comment about synchronization that we see in the brain of different pools of neurons, whether there is, you think there is a mechanical basis of this synchronization?
Yeah, so both are good questions. So I think that the softness helps to isolate individual synapses and keep them as a discrete mechanical unit. Obviously, they've got other like abstracts and other stuff talking to it, but it means that they can, that can be a discrete mechanical computational device because it's in control of all of the settings. Things where it gets stiffer would be an easy way to imagine how that could lead to disruption or dysfunction of this. So even like the Alzheimer's, and maybe they stiffen the matrix gets stiffer, or there's some clogging up of it. So yes, I'd say the softness is an advantage. It was for a long time thought to be a disadvantage for mechanical transduction, but if you imagine it as a discrete mechanical unit, then yes, I think it's an advantage. And then yeah, the second question is like so this is all to be tested, obviously, but it's start to imagine that if the probability of each synapse or each firing is coordinated, if the whole cell is behaving as a mechanical computational computer, and if you're dialing up and down different synapses activity, so if you stimulate one, you might tune up another, or it might be able to send this to very discrete circuits. There might be if you activate one synapse, you will stimulate a different circuit if you activate another synapse, even though they're on the same neuron, just because of how the cytoskeleton's wired up and where the location of the enhancers and dampeners are, or something like that is what I'm trying to imagine. And the state of all of it, because it'd be like a mechanical computation in one cell, which would be in a circuit, which would be connected to another circuit. So there'd be a huge amount of yeah, it's all to be worked out, I'd say. And I don't think that answers your question at all, but it would be, I would imagine that within certain circuits, they would be preferentially stimulated once you stimulate. There might be entry points into those circuits, or like, yeah. Sorry, that's a terrible answer, but I think, yes, it's complicated. But that does happen. I mean, as you're saying, that works out that those circuits are being stimulated anyhow. So this would just be that whatever's happening to do what we know already about neuro would be updating these coding activity during that process which is going on, which at the minute seems like the probability of neuronal release vesicular release seems quite magical, I'd say. It's like yeah, I don't know. I'm just shut up hanging myself here.
So can I ask you a question? I mean, just like more maybe science fiction about this, but yeah. According to to your thoughts on the theory, what would be a way of increasing memory formation through mechanical manipulations or something like that?
I don't know is the short answer, because I think that's really complicated, but maybe I'd imagine you'd be able to read this before you could write it, because it might be that we wire up beautifully, like all of our fingers and everything works. So there's a body plan. So maybe the brain's equally logical, and maybe there's rules with how it's written, and it might be indexed depending on how you learn to speak or whatever. But there might be that there's simple rules to how the information is written into the cortex, is what I would speculate. And it might be that it's possible to read that by reading the current back to it, but we don't know the operating system. So until you know that, or until you know what the coding is, it's quite hard to extract these at the minute electrochemical signaling or patterns of ones and O's. Until you know what the actual underlying coding of that is, it would just be yes is the short answer. I don't know how to write it at the minute or how to improve it. You might be able to protect it by helping to modulate the mechanical environment, or treat things to stop the mechanical environment stiffening up, or to keep them supple, like when you do exercise with your body, maybe. But yeah.
Thank you. I do have a couple of questions about the more molecular on tailing. So the first question is, are these domains open sequentially and the sequence is always the same, or it's more randomized?
It's definitely not random, and it's sequential. There's a couple, like there's some which have got very similar mechanical responses, so there might be one or the other one would go, but yes, in broad strokes, you could predict which one would go next. And what we've also got is we've got the vinculins binding, and we've also got phosphorylation events which lock them in a specific state. So like we've just published on the CDK1 phosphorylating tailin, it changes the order that the switches go. So you can start to imagine that it depends on other inputs. So we start to even imagine like logic gates, where if you've got an active kinase and this amount of force, you get this response. If you've just got the kinase, so etc., you can start to predict. And then because the integrin tailin actin is at the top of all of the mechanotransduction chain through the adhesions, that those patterns you determine at the top of the cell, at the start of that process, then determine how the whole cell behaves. So yes, I think it's quite.
Yeah, so correct me if I'm wrong. I'm going to make a couple of assumptions now that I'm not sure about. But you said that the domain R7 recruits microtubules, and I'm assuming that microtubules are recruited for disassembly. So does this mean that there is a threshold force for which disassembly of the focal punct or focal complexes are initiated?
Yes, so the reason that recruits microtubules is that it's got a binding site for a protein called CANK, and in certain mechanical scenarios, CANK goes right to the goes to the tailin, but for some reason it only goes to tailin on the edge of the adhesions. But it makes basically a complex which captures and stabilizes microtubules as they grow out. So they grow to the edge of the cell. If they encounter these clasps and kinesin complex, it stabilizes and holds it there for a long period. The classical view in terms of migration is that turns over the adhesion complexes, yes. But that's in a more stable environment, like in a layer of cells or in an organ. It might be that it's holding them there a long time. And it might be that actually wires it up so the microtubules persist, go into that site. So right, yeah, because that was what I was assuming, and I wasn't sure, because that could also be the same mechanism by which then the synapse gets potentiated. So it recruits the microtubule, and therefore more molecules are transported actively to that synapse as compared to others. Yes, definitely, and we don't know. One of the things we're trying to do at the minute is to find the neuronal tailin ligands, because there might be new ones and new players in this. Because, but one of the cool things is, and one of the being biochemists, we can put a single point mutant in R7, so that microtubules no longer go to adhesions at all. Period. And that's been quite useful for our work in the cancer cells, because we can physically disconnect microtubule targeting of adhesion sites. So the capacity to do that with all the tailin ligands and to change the order the switches go means that we've got this ability to reprogram this quite well. Maybe not in our brains, not to increase our learning, like Miguel said, but in terms of to work it out on a cell basis or in a synapse in a culture, we can certainly take this apart.
Fantastic. Thank you, Ben. Thanks a lot. So if there are no other questions, then I